Hi students, welcome to the lecture on hormones and enzyme. I'm Carolina Santiago and here is my email address. Do contact me after the lecture if you have any questions. I have divided this lecture into two parts. So in part 1, we will be looking at enzymes. So here are the learning outcomes for this lecture which are related to enzymes. There are 4 learning outcomes related to the topic enzymes. At this point, you may want to pause your video to read through these learning outcomes. The learning outcomes are important because your exam questions are blueprinted to these outcomes, meaning the questions that we author are guided by these outcomes, and these outcomes also serves as a guideline for your revision. Let's start our lecture with a warm-up question. So hexokinase and glucokinase are two enzymes that catalyze the same reaction. However, glucokinase will only catalyze the reaction when glucose concentration in the blood is higher than normal. What property of glucokinase explains this phenomenon? So there are 4 options here for you to choose. So hold on, let's see whether you can answer this question at the end of the lecture. Maybe some of you already know the answer, which is excellent, but let's go through the content of this lecture and we will revisit this question once again. So what is an enzyme? Enzyme is a biological catalyst and most of the time it's protein, and it increases the rate of chemical reaction in a cell. So here is an example of enzyme named sucrase. You can see that it has an active site which is complementary to its substrate sucrose, and the binding of enzyme with its substrate creates enzyme substrate complex, which goes through chemical reaction such as hydrolysis in this case, and finally release the product glucose and fructose. After releasing the product, the enzyme is free and it could bind to another substrate. There are 2 models which explain the enzyme substrate interactions. The first one is lock and key, where the active site is a perfect fit for the substrate and does not require any changes for it to bind. And this is why we call it lock and key, because they are a perfect match. The second model is induced fit. Alright, so in induced fit model, the active site is almost complementary to the substrate, but when it binds to the substrate, the enzyme goes through a conformation changes to fit the substrate better. This is a much more widely accepted view in regards to model of enzyme substrate interaction. Right, so many times we hear that enzyme lowers activation energy. So what is activation energy? Let's go through this slide. So this is a basic chemical reaction where you have your substrate and then it goes through a reaction producing the product. The reaction is also reversible, but bear in mind one direction may predominate the other. Now let's look at the second equation here, where you have enzyme plus your substrate. It goes through a reaction and forms enzyme substrate complex. So enzyme substrate complex is a transition state, which is a high energy state, all right? And the reaction continues producing the product and releasing the enzyme back so it can be used again. Now, let's look at this graph here. Okay, so this enzyme substrate complex or the transition state is a high energy state, yeah? So looking at the graph here, you can see that without the enzyme, for the molecules to reach the transition state, it requires a very high activation energy. Whereas with enzyme, the green line here, you can see that the activation energy required is lower. Alright, so in real life, only a few molecules have enough energy to reach the transition state. Alright, therefore, it is very unlikely for any chemical reaction to occur in cell without the presence of enzyme. Okay, now what is this activation energy? So to give you an analogy, imagine if you have a ball sitting in a hole on the top of the hill. For the ball to go zipping down the hill, it has to come out from the hole that it is sitting in. So the initial push that you give to the ball to come out from the little hole is the activation energy. So meaning it is the energy required for the chemical reaction to start or to progress. So now let's look at how exactly enzyme lowers activation energy. So there are two ways in which an enzyme can lower the activation energy. So the first way is through the internal rearrangement. Okay, for example, you have an enzyme and this is the active site, which it could accommodate 2 amino acids, and then these 2 amino acids are being joined together here, all right, to produce a dipeptide product. Okay, so the rearrangement helps the formation of the chemical bond required to form the product. The second way which it could lower the activation energy is enzyme can also stretch the bonds in the substrate, making them unstable. Therefore, the bonds could be broken. Alright, so you can see here, this is the enzyme, and here you have the substrate, and it goes through some stretching mechanism here. Therefore, the bonds between the molecules in this substrate can be broken, and eventually this will break the substrate into smaller units. Now let's look at factors affecting enzyme activity. The first one is temperature. So here is your enzymatic activity, here is your temperature. Alright, and you can see as you increase the temperature, the activity of the enzyme increases and it reaches the maximum at the temperature of 37 degrees Celsius. Alright, since we are talking about enzyme in relation to chemical reaction in a cell, the optimum temperature is 37 degrees Celsius. Anything above 40 degrees Celsius will denature the enzyme because they are basically protein, and that's why you see the activity becomes low or decreases. Next is pH. And you can see for this particular enzyme, the optimum pH is about 5. Okay, so some of the enzyme may have an optimum pH of 8 and some of it may have about 4 or even lower. So this is based on what type of reaction the enzyme catalyzes and where it is present in the body. Alright, so if you're talking about enzyme that is secreted together with the gastric juice in the stomach, it may have an optimum temperature which is more acidic, right? Now let's look at the third factor, which is substrate concentration. So you can see that as you increase the substrate concentration, the rate of reaction or the enzyme activity start to increase as well. But at one point, this line becomes flat, okay? So this is because, the concentration of the enzyme, okay, becomes the limiting factor. No matter how much you increase the substrate concentration, at one point, if all the enzymes are occupied, okay, then you would not see any increase in the enzyme activity because it has reached its maximum rate of reaction. So this diagram is just to show you the differences in terms of optimum pH. For example, pepsin, an enzyme which you find in stomach, has an optimum pH of 2, whereas trypsin has an optimum pH of 8 and you find it in small intestine. So now it's time for us to do a recap question. Here you see two graphs, okay, graph A and graph B, okay, and how will an increase in enzyme concentration affect the rate of reaction? All right, so if you look at the first graph, you see as you increase the substrate concentration, the graph indicates that the reaction rate increases, but after a point it starts to curve, all right, forming a hyperbolic curve. In B, you can see as you increase the enzyme concentration, you get a straight exponential increase in the rate of reaction as well. Why is that so? So the answer would be increasing enzyme concentration will speed the reaction as long as there is substrate available to bind. So as you keep increasing the enzyme concentration, the rate of reaction will increase as long as there is substrate available. Okay, so once all of the substrate are bound, the reaction will no longer speed up since there will be nothing for additional enzyme to bind. So that's why you get a straight line. Whereas with substrate concentration, as I explained in the earlier slide regarding factors affecting the enzyme activity, at one point all the enzyme is occupied with the substrate. Okay, so further increasing the substrate concentration will not increase the rate of reaction. Therefore, you start to see the line slowly becoming flat. All right. Now let's look into Michaelis-Menten enzyme kinetics. So this is one of the important part in the topic of enzyme. Okay, basically this Michaelis-Menten kinetic model explain how an enzyme catalyzed reaction depends on concentration of enzyme and its substrate. Okay, so let's look at the equation here. So you have substrate, enzyme binding together, forming enzyme substrate complex, and this reaction here is reversible. But once the reaction proceeds to form product and releases the enzyme again, we assume at this point the reaction is irreversible. All right. So here, this is basically the Michaelis-Menten equation, and not to worry about this, you are not required to memorize this equation, all right? But it's just good to show as a part of this topic, all right? So in Michaelis-Menten enzyme kinetics, all right, there are two orders. First is the first order. Second is zero order. Both of these order explain how the rate of reaction is affected by the substrate concentration. Okay, in the first order, as you look at the graph, okay, so here this y-axis here represent the velocity or rate of reaction. The x-axis here represent the substrate concentration. You can see when the substrate concentration is increased, a rapid increase in the rate of reaction in a linear fashion. All right, so in this graph, we can say that the rate of reaction is affected by the substrate concentration, and this is known as first order kinetics. Now let's look at the second graph. Okay, in the second graph, you can see that this rate then start to plateau as you increase the substrate concentration, and any increase in substrate concentration at this point has no effect on the rate of reaction. This is because all the active sites on the enzyme are saturated with the substrate. So you call this order as zero order kinetics. All right. So this is a very important feature of the Michaelis-Menten enzyme kinetics, which is at the initial point, the rate of reaction depends on the substrate concentration, but as you increase the substrate concentration, the rate of reaction no longer depending on the substrate concentration. Now, there are two important terms within the Michaelis-Menten kinetics. Look at this graph. So here on the y-axis, you have the velocity or the rate of reaction. On the x-axis, you have the substrate concentration. Okay, so Vmax is the maximum rate of reaction, and it is the point where all the enzyme's active sites are saturated with the substrate. So this is the maximum rate of reaction that you can get with this particular enzyme. Now, there is also Km. Km is known as the Michaelis constant. Numerically, Km, as you see on this graph here, it is the point where the substrate concentration corresponds to half of the Vmax. Okay, so it is the substrate concentration needed to reach half of the Vmax. So if this is 100, you can imagine this is 50. All right, and Km, besides representing the substrate concentration to reach half of the Vmax, it also represents the affinity of the enzyme has for its substrate, meaning how well the enzyme binds to a substrate. So if you have a small Km, meaning the enzyme has high affinity to the substrate, okay, because you only need a small concentration or a low concentration of the substrate to reach half of the Vmax. If you have a large Km, that means the enzyme has low affinity to the substrates, hence it need more concentration of the substrate to reach half of the Vmax. If you remember the graph from the previous slide, you will note that at one point the lines start to become flat and form a hyperbolic curve. When this happens, it's hard for us to determine the Vmax. Alright, therefore we have this Lineweaver-Burk plot whereby we plot inverse of the reaction rate against the inverse of the substrate concentration. So basically you plot 1 over velocity and 1 over substrate. And when you plot the graph this way, you will get a straight line. And the point at which the line intercepts on the y-axis is represent 1 over Vmax. And the point at which the line intercepts with the x-axis, it represents negative 1 over km. So from this graph, we can determine the km value and the Vmax value. So we have looked at introduction on enzyme. We have looked at factors affecting enzyme activity, and then we have looked at enzyme kinetics. Now let's look at enzyme inhibition. So an inhibitor is a substance that decreases the velocity of enzyme catalyzed reaction. Basically, it inhibits the enzyme's action. And in essence, you can divide the type of inhibition to irreversible and reversible. So irreversible inhibition is when a substance can bind to the enzyme covalently. A covalent bond, it's a strong bond and it's hard to break that bond. So these binding become irreversible. And the inhibitor, because it has bound to the enzyme tightly, it does not dissociate, alright, therefore it inhibits the action of an enzyme. So an example is the antibiotic penicillin, where it could bind the transpeptidase enzyme in the bacteria. This enzyme is responsible for formation of bacterial cell wall. So by inhibiting this enzyme, we can prevent the bacteria from multiplying and eventually killing the bacteria. Alright, so the next type of inhibitor is reversible. So these kind of substance, they bind to the enzyme through a non-covalent bond. So they do not go through any chemical reaction with the amino acid found at the active site of the enzyme. Alright, so it can be removed from the enzyme provided you increase the concentration of the substrate. So let's look at the diagram here. So here you have HMG-CoA reductase enzyme, which is the enzyme involved in the synthesis of cholesterol. Alright, and you have a drug here. Alright, this is Pravastin. It's a type of statin drug that is designed to reduce the synthesis of cholesterol for individuals that have high blood cholesterol level. So this drug has got similar structure to the substrate of the enzyme, which is HMG-CoA. Therefore, this drug could bind to this enzyme and inhibit its action. Eventually, this will result in reduced level of cholesterol synthesis in the body. There are several types of reversible inhibition. Let's look at the types. So here is your enzyme and here is the active site and this is the substrate. Okay, this is how the normal binding occurs. Now you have competitive inhibition. In competitive inhibition, the competitive inhibitor binds to the active site and prevents the substrate from binding to the active site. So there's a competition to bind to the active site between the inhibitor and the substrate. Therefore, the name competitive inhibition. Then you have non-competitive inhibition. Non-competitive inhibition where the inhibitor molecule binds to other than the active site, as what you see here. This binding changes the structure of the active site so that it is now no longer complementary to the substrate. So the substrate cannot bind to the active site. Then you have uncompetitive inhibition. Uncompetitive inhibition is when you have the inhibitor molecule binding to the enzyme substrate complex. When it binds to the enzyme substrate complex, it prevents the product from being released from the enzyme. So we call this uncompetitive inhibition. Moving on, let's look at enzyme inhibition and their effects on Vmax and Km. Before I explain further, let's recap on Km. Km represent the affinity of an enzyme to its substrate, meaning how well the enzyme binds to its substrate. Let's say if you have a low Km value, that means the enzyme has got high affinity for the particular substrate. And when you have high Km value or a big Km value, that means the enzyme has got low affinity to that particular substrate. Alright? Now let's look at competitive inhibition. So in competitive inhibition, this is the graph when you plot with and without the inhibitor. In competitive inhibition, the inhibitory molecule binds to the active site. So when it binds to the active site, it reduces the affinity of the enzyme to the substrate. Therefore, you see an increase in Km value. Whereas the Vmax is unaffected, because sooner or later, as you keep increasing the substrate concentration, the substrate can overcome the inhibitory molecules and finally the enzyme will reach its Vmax. Moving on, let's look at uncompetitive inhibition. So this is the graph as what you can see here, and from this graph we can deduce, alright, the Km and Vmax value in terms of whether it increases or decreases, and you can see both Km and Vmax is reduced. Okay, so it is a bit odd, okay, but note here the Vmax is reduced. Okay, because in uncompetitive inhibition, the inhibitory molecule or substance bind to the enzyme substrate complex. Okay, so the number of available enzyme is reduced, therefore the maximum rate of reaction is reduced as what you see here. However, note that Km is also reduced. Why is that so? Okay, so the Km value decreases because the enzyme affinity for its substrate increases. Because what happens is when there is a decrease in the number of free enzyme, the available enzyme that could function will develop a greater affinity for its substrate. Therefore, you see the Km value being reduced. So increased affinity to the substrate for the available enzymes. In non-competitive inhibition, as what you see in this graph, Km is unaffected. Okay, this is because the affinity of the enzyme does not change. Yeah, because the inhibitory molecule is not binding to the active site. Alright, therefore the affinity remains the same. However, the Vmax is reduced because the number of functional enzymes that can carry out the reaction is reduced because they are bound by the inhibitory molecule. Therefore, the substrate cannot bind to the active site. Okay, so you see a reduction in the Vmax. Next, let's look at regulation of enzymatic activity. So, its activity of enzyme is an ongoing reaction without a regulation? No. There are regulatory mechanisms, all right? One of it is allosteric regulation, where a molecule can activate or inhibit the enzyme action. So if you look at the picture here, this is allosteric inhibition, where the inhibitor binds to other than active site known as allosteric site, and that changes the structure of the enzyme, especially its active site. The substrate cannot bind. Yeah, so this is an allosteric inhibition. Next, you have allosteric activation. So this is when a molecule, we call it an activator, it binds to an allosteric site, which simply means a site different than the active site, and that results in changes of the enzyme structure, permitting it to bind to the substrate. So apart from allosteric regulation, you also have cofactor, which are a group of molecules that regulate enzymes activity. They are non-protein molecules and they help enzyme to achieve an active state in order to catalyze a chemical reaction. When they attach temporarily to the enzyme, you call them co-substrate. When they attach permanently, such as the inorganic ions, you call them prostatic group. And if they are organic molecules, you call them coenzymes. Okay, so there are different names based on the type of molecules and based on how they attach to the enzyme. The most common source of coenzyme are dietary vitamins. Okay, for example, vitamin C is the coenzyme for several enzymes that take part in the building of protein collagen. Yeah, right. Next, you have feedback inhibition, which is a mechanism that regulates the enzyme activity. Okay, so look at the picture here. This is a glycolytic pathway. Alright, so you can see that there are many products here along the pathway, and each of this reaction here involves an enzyme. Okay, in feedback inhibition is when you have an end product that could inhibit the key enzyme involved in that particular pathway, and this stops the pathway from producing more end product. Okay, so in this example, this is glycolysis pathway. The end product is pyruvate and ATP, and the presence of ATP as an end product will inhibit one of the key enzyme in the glycolysis pathway, which is phosphofructose kinase enzyme. Next, you have feedback repression. In feedback repression, the accumulated end product function as a repressor, meaning they could bind at molecular level to the gene, known as the operator, which encodes for the synthesis of the particular enzyme and stop the enzyme synthesis. Just now, in feedback inhibition, the end product inhibits the enzyme itself. Now you have the end product inhibits the operator or the gene that encodes for the synthesis of the enzyme. This is known as feedback repression. Lastly, you have covalent modification. Covalent modification is when you add a group through covalent bond. It can make the enzyme active or inactive. Alright, so the most common covalent modification is adding and removing phosphate group. So here is an example of glycogen phosphorylase. It is inactive when you don't have phosphate group, but when you add phosphate group to the enzyme, it becomes active. And that will bring us to the end of part 1 enzyme lecture. After this, you have a quiz with a number of questions, and we will look at the warm-up question as well. Make sure you try this question before you watch part 2 of this lecture. Thank you. Okay, so this is the warm-up question which I shared at the beginning of the lecture. You can pause the video for a while to attempt to answer this question. Right, so which of the following is the correct answer for this question? So if you answer low affinity for glucose, that is the correct answer. So hexokinase and glucokinase, they both catalyze the same reaction, which is phosphorylation of glucose. Okay. When the concentration of glucose in the blood is high, hexokinase will be the first enzyme to carry out that reaction. Yeah, that is because hexokinase has high affinity to the glucose molecules. Okay, so hexokinase will be binding to all these glucose molecules and will catalyze the reaction. Okay, at one point all the enzyme is occupied with the reaction, but you still have excessive amount of glucose in the blood. That is because here in the question they have mentioned that glucose concentration in the blood is higher than normal. So what happened to these excessive glucose molecules that are still in the blood while all the hexokinase is occupied? So this is when glucokinase will come into action and it will catalyze the reaction for the remaining or the excessive glucose molecules. Okay, and glucokinase has got low affinity for glucose, so it will bind to the remaining glucose molecules. So I hope that explains the answer. So all of the other options A, C and D has got nothing to do with the question really. Yeah, in the question they are not talking about inhibition or the location or the molecular weight. Yeah. Let's move on to the next question now. Right, now let's move on to the subsequent question. Question 1, you have a graph showing gastric protease and intestinal protease. Alright, and you can see from the graph both of these enzymes have got different optimum pH. So the question is asking what will happen if you mix both of the enzyme together with their substrate in a single test tube. So you can pause the video and try to answer first before we discuss the feedback. Right, okay, so what is the answer? So A, the gastric protease would be active if the pH of the mixture was basic. So this option is not clear what they mean by basic. So this is a wrong option. The intestinal protease would be more active than gastric protease at pH 4. That is also incorrect because intestinal protease has got optimum pH about 8 here. So that is incorrect. Both enzymes would exhibit some activity at pH 5. So let's look at pH 5. Yeah, it is not great, but you can see that there is an increase in activity. Yeah, for both type of protease, gastric and intestinal. So this is the correct answer. Okay, because you can see some activity at this pH. The last option, gastric protease, would be more active than intestinal protease at pH 6. That is incorrect because gastric protease, you can see its optimum pH is acidic. Yeah, so you can't expect it to be more active at pH 6. All right, now let's move to the next question. For the next question, we have a table with data. So here you have the substrate concentration, and here you have the velocity or rate of reaction. So if you plot a graph, the substrate concentration will be on your x-axis and the velocity will be on your y-axis. So the question is, what is the approximate km for this reaction? To answer this question, you need to know what km is. Right, so you may want to pause for a couple of minutes to answer this question, and then we can look at the feedback. Right, okay, so km is the substrate concentration which correspond to half of the Vmax. Okay, so if we want to find this km value here, first of all we need to find what is the Vmax for this reaction. So from the table, this is where you have the velocity, and the maximum rate of reaction is 280, because after that, even when you increase the substrate concentration, it is about the same, 279. So we can say that 280 is the Vmax. So half of 280 would be 140. So this is your half of the Vmax. So Km is the substrate concentration at half of the Vmax. So half Vmax is 140. And what is the Km or the substrate concentration at this point? Is 2 times 10 to power of negative 5. Okay, so the correct answer would be A. Right, so this is the last question for the enzyme part. Read the question carefully. The question has a diagram where you can see that there are 3 amino acids, starting with amino acid 1 and then to 2 and then to 3. 2 enzymes are involved, V and X. So you can think of it as a pathway or a part of a pathway. Alright, so now the question is asking, when an excess amino acid 3 is added to the system here, the rate of reaction is reduced. What is the cause of this? At this point, you can pause the video and try to answer on your own first before we look at the correct answer. Okay, now let's look at the answer. A, end product inhibition. Is this the correct answer? Yes, this is because amino acid 3 is the end product of this particular reaction. And when you add an excess of amino acid 3, you can see that the rate of reaction is reduced, meaning there is an inhibition of the enzyme involved in this pathway. So this is a sort of end product inhibition. When the end product has exceeded certain level, it will start to inhibit the enzyme involved in the pathway. Alright, so option B, enzyme denaturation. There is no mention about changes in pH or temperature here. So we can safely say this does not involve any enzyme denaturation. Excess substrate inhibiting the enzyme. So the substrate for this reaction is starting off with amino acid 1 and 2. Okay, and we are not adding any more of 1 and 2 amino acid into the system. So this is incorrect. Positive feedback is also incorrect, because positive feedback means it will enhance the reaction where you will see increased rate of reaction, not reduce. Yeah, so the correct answer is A. Right, so I hope the example of questions have given you some insight, yeah, about how this topic can be tested in the exam. Right, thank you very much, guys.